Many studies have shown that the onset of fever with malaria is highly correlated with a dramatic rise in serum levels of the inflammatory cytokines interleukin-1 and tumor necrosis factor alpha. So hopefully you recall from our immunology lectures that interleukin-1 and TNF-alpha are released by activated macrophages, which are the phagocytic cells that are trying to deal with all the cell debris resulting from the rupture of erythrocytes. And this cleanup process is occurring in the spleen and liver. So as the macrophage becomes activated and releases these cytokines, they travel in the bloodstream to the hypothalamus of the brain. And the hypothalamus then triggers rapid rise in fever, the contraction of muscles that occurs in an effort to try to generate some of that temperature increase is why we have chills that precede fever. So huge rapid rise in fevers of 104 degrees Fahrenheit, 105 degrees Fahrenheit, very typical. The patient feels terrible, which is malaise. They have no appetite, anorexia, and the other symptoms that occur during the paroxysm are also triggered by the stimulation of the hypothalamus by TNF-alpha and IL-1. So it's been shown that the severity of that paroxysm can be directly correlated with the levels of TNF-alpha and interleukin-1 produced. And that's going to be correlated with the extent of red blood cell lysis. Anemia that results during malaria clearly occurs because of large-scale destruction of red blood cells, but also from the phagocytosis of infected and uninfected erythrocytes, which occurs at least in part because of infected red blood cells' tendency to clump together. TNF-alpha, in addition to triggering the hypothalamus to cause the symptoms of malaria, also inhibits hematopoiesis, blood cell formation. So you have vascular thromboses where cells are being aggregated and removed from the circulatory system, and then you have cell lysis, and then you have a suppression in hematopoiesis, and so collectively the amount of anemia can be extreme with some species of malaria. Another factor is that a lot of the iron that is present is tied up in the form of hemozoin, and so that hemozoin pigment is building up within tissues and organs, and the iron is not efficiently recycled for use by newly synthesized hemoglobin. Another corollary with malaria is organ pigmentation. So hemozoin and other byproducts of hemoglobin do build up in tissues. With severe malaria, at autopsy, the spleen and the liver can look black. The white matter of the brain can look gray. And again, this is due to pigmentation, deposition of hemozoin, and phagocytosis of large quantities of pigment by fixed macrophages in these tissues. Vascular occlusion, which, as we said, is most severe in Plasmodium falciparum infection, is most serious when it impacts the brain and causes cerebral malaria. And this is occurring because of the parasite antigens that are expressed in the knobs of the erythrocytes. So the parasite itself is inducing these knobs, and keep in mind there are parasite proteins within those knobs. The presence and alteration of the surface of the red blood cells causes the charge characteristics of the erythrocytes to change. So normally our erythrocytes are negatively charged, largely because of sialic acid that's present. And of course, like charged cells are repelled from each other, and this helps to keep our red blood cells separate from each other and flowing nicely within our bloodstream. So when erythrocytes become infected by falciparum, Plasmodium falciparum, We have knob formation, we have alteration in charge, the red blood cells become more neutral, and this causes the cells to become sticky. Once we have a buildup of these red blood cells in capillaries and postcapillary venules, we can get complete blockage of the blood supply, and this can cause local hemorrhage and necrosis, and of course anoxia, which is followed by local cell death and convulsions. In the brain, cerebral anoxia often occurs, and it's a very common cause of death with falciparum malaria. With all forms of malaria, an enlarged spleen is a hallmark of infection. Keep in mind the spleen is there to filter our bloodstream, and it's very important in our defense against bloodborne pathogens. And so the spleen does respond to this onslaught, really, of infection. And because of the lymphocyte activation that occurs in the spleen and an influx of lymphocytes into the spleen can become dramatically enlarged. And in fact, from a clinical point of view, the extent of splenomegaly in villages where malaria is endemic is often used by clinicians as a measure of malaria prevalence in a particular season. Okay, so now a little bit about the epidemiology of malaria. Not surprisingly, the distribution of malaria is determined by the distribution of the Anopheles mosquitoes. And there is growing concern that climatic changes that have occurred that are leading to global warming will allow for an expansion of Anopheles mosquitoes, and not just Anopheles, but other arthropod vectors as well. There is an interesting correlation with age and susceptibility to malaria in endemic areas. So just want to be clear about that. So in areas of the world where malaria occurs regularly, children between the ages of 6 months and 5 years are most susceptible. Younger infants are protected by congenitally acquired immunity due to maternal IgG. So by the time a woman becomes pregnant, no doubt she has had malaria multiple times and has circulating antibodies that are protective. And these maternal IgGs cross the placenta and protect the infant in the first few months of life. But as the maternal IgG disappears and as the mother begins to stop nursing between 6 months and 1 year of age, the protection is lost, and children that have just weaned are incredibly susceptible to infection. And in areas of high endemicity, children under the age of 5 experience the greatest suffering and the highest rates of mortality. Now, in endemic areas, a form of clinical immunity does develop with reinfection, and we're talking about many, many times of being reinfected. There is some evidence that the immunity is cross-protective, so if you've had Vivax, it can confer partial immunity to falciparum and vice versa. The important point is that this is not sterile immunity. Sterile immunity occurs when the adaptive immune response and the memory response is sufficient to prevent you from being reinfected. So sterile immunity never develops with malaria. And instead, what you see in endemic areas is premunition. Premunition is protection from superinfection or reinfection, and it only occurs if you are harboring Plasmodium at the time. So immunity builds with age and re-exposure and with individuals who continue to harbor low levels of parasites in their bloodstream. It turns out, sadly, if you cure an individual of malaria with drug treatment, the premunition is lost. Now, in contrast, in areas where malaria does not occur, so if you had a new outbreak of malaria in an area where it was not previously endemic, you would see that all age groups are affected equally because there is no clinical immunity in the population. There's a very interesting example of balanced polymorphism in human genetics, and this is related to the resistance that's conferred due to the sickle cell trait. So the condition known as sickle cell anemia is caused by a genetic mutation, and that genetic mutation resulted in the substitution of one glutamic acid by a valine. So an acidic amino acid in the structure of hemoglobin is replaced by a basic amino acid valine, and that change is very significant with regard to the structure of the hemoglobin protein. Now, in the homozygous recessive condition, historically, that condition has been lethal. So an individual that has sickle cell anemia typically did not survive to reproduce. In today's world, with treatment and supportive care, these individuals are living a much longer life. But ordinarily, a homozygous recessive lethal condition would be removed from the population over time because of its lethal effects. But clearly, malaria has provided selective pressure that favors heterozygotes. Because individuals that are heterozygous for the sickle cell trait and homozygous individuals do not get malaria. The sickle cell hemoglobin is just not compatible physiologically with maintenance of malarial infection. The treatment of malaria, at the present time, there is no vaccine available for malaria, and that's despite considerable investment of time and money for decades. The other big concern with regard to malaria is the emergence of drug-resistant strains of malaria, as well as a shortage of chemotherapeutic agents for the future. The use of quinine to treat malaria dates back to the 16th century. The mechanism of action is not completely known even today, but it's thought that quinine and quinine derivatives interfere with the ability of Plasmodium to metabolize hemoglobin. Quinine affects the erythrocytic stages of Plasmodium only and has no effect on gametocyte stages or liver stages, exoerythrocytic stages. Over time, quinine use fell out of favor due to its severe side effects and due to quinine resistance in Plasmodium populations. Now, there were derivatives of quinine that were developed and began to come into use in the late 1940s, chloroquine and primaquine to be exact. Chloroquine, like quinine, is primarily effective against erythrocytic stages and is used today in areas where chloroquine resistance has not yet developed. In many parts of the world, we cannot use chloroquine because of the spread of chloroquine resistance. Now, primaquine, which was developed at about the same time, is the only one of the quinine family of antimalarials that's effective against the liver stages and against the hypnozoite stages of relapsing malaria, Plasmodium vivax and Plasmodium ovale. But, unfortunately, Plasmodium resistance to primaquine has also arisen and is a major factor that limits widespread use. The ideal situation with a relapsing form of malaria is to give chloroquine and primaquine to eliminate the infection. There are a variety of newer drugs. Methylquine was developed in the 1980s. The most promising had turned out to be artemisinin, or qinghasu, which is an ancient Chinese remedy for malaria. Artemisinin was a tremendous find because it was useful in chloroquine, primaquine resistant areas, but now artemisinin resistance is spreading rapidly, and it's really quite alarming. So to try to sort of slow down resistance to malarial drugs in Plasmodium, typically now clinicians try to use a combination therapy where they are exposing patients to at least 2 different anti-malarial drugs. And one example of that is a drug known as Malarone, which is actually a combination drug. Now, so we don't have very much in our arsenal to protect against malaria. The drugs that have been developed are rapidly becoming useless as resistance spreads in the Plasmodium populations. And another huge problem with malaria is that mosquitoes are emerging that have insecticide resistance. And together, these problems make malaria such a formidable enemy. All right, the immune response to Plasmodium infection is complex because Plasmodium has such a complex life cycle. Plasmodium has evolved a number of ways to avoid the host immune response. They use a hide-and-seek strategy by having both intracellular and extracellular stages within the vertebrate host. They take advantage of antigenic variation, not to the extent that we saw with African trypanosomes, but they still can utilize antigenic variation, especially of immunodominant surface antigens, almost like a decoy strategy. And then, of course, the complexity of the life cycle also works to their advantage because each stage is antigenically distinct, and there is not very much cross-protection between the different stages. However, protective immune responses clearly do occur. If you calculated the potential for replication in these organisms, it is clear that death would occur in a matter of a few days in the absence of an immune response. So the immune system does, if the patient survives, it does protect against widespread parasite replication, dissemination. But in a way, the immune system actually works to the advantage of the parasite by by not eliminating it and keeping it at low levels in the host. Again, to reiterate, sterile immunity to Plasmodium does not develop. There are innate immune mechanisms that protect us against malaria. It's predominantly based on macrophages because this is a vector-borne disease. Mosquitoes are actually inoculating the parasites into the bloodstream, so our normal anatomical barriers are not so helpful. Macrophages, though, can remove the stages of Plasmodium, the extracellular stages of Plasmodium, and they also have the enormous job of cleaning up dead erythrocytes, dead and damaged parasites, hemozoin, and hemoglobin byproducts. So macrophages have a really busy job when malaria is taking place, and the recognition of both PAMP and DAMP signals associated with malaria are important in leading to macrophage activation. There hasn't been a huge amount of work done on this yet, but we do know that toll-like receptor 9, toll-like receptor 2, and toll-like receptor 4 are all important. And when these toll-like receptors engage PAMP and signal transduction occurs, that triggers the activation of macrophages and the release of pro-inflammatory cytokines. Of course, these cytokines are essential in getting the response going, but as we've learned, they also contribute greatly to the pathology. In terms of adaptive immune mechanisms, as you might expect, antibody-mediated responses should provide some protection against those extracellular stages like the merozoite and sporozoite stages within the mammalian host. And we know that individuals in endemic areas who are exposed and re-exposed to Plasmodium do develop anti-Plasmodium antibodies, and clearly those antibodies are having at least some effect. However, the efficacy of the antibodies is limited because the parasites do have the intracellular life cycles, and because these extracellular stages are really transient, so the antibodies and the mechanisms that antibodies use to protect against extracellular stages have to act really efficiently. The sporozoite stage, which keep in mind is only in the circulation for 30 minutes or less. Only one sporozoite has to make its way to the liver to establish infection. The sporozoite is covered with a protein called the circumsporozoite protein. So it covers the entire surface of the sporozoite, and it's highly immunogenic. It does stimulate a strong antibody response. However, the sporozoite has the ability to shed that circumsporozoite coat, and once it's shed, it's basically a decoy that allows antibody to bind to the shed protein, allowing that sporozoite to slip into a liver cell to establish the exoerythrocytic phase. Each Plasmodium species, and even different strains of Plasmodium, are antigenically distinct in terms of the circumsporozoite proteins. Another factor is that even if we make antibodies against Merozoite and Sporozoite stages, those antibodies don't necessarily block the ability of those stages to infect erythrocytes. And part of the reason for that is that the proteins the parasite uses for attachment and invasion are packaged inside the apical complex, and specifically within micronemes and rhopteres. And these proteins are only released after the merozoite physically binds to the erythrocyte. So they're very directed onto the surface of the erythrocyte, and they don't end up stimulating very much of an antibody response. Now, we have shown that antibodies can potentially serve as opsonins that coat these stages and enhance phagocytosis. There's also some evidence that ADCC mechanisms can be used to kill extracellular stages, and even antibody complement-mediated lysis can occur. But again, we see clinical immunity developing in endemic areas, but this is largely a premonition phenomenon where the person is continuing to harbor low levels of the parasite. Just to remind you that the humoral response to malaria is supported by a B lymphocyte response that's supported by T helper 2 CD4 positive T cells. T helper 2 cells promote B lymphocyte activation, B lymphocyte growth, division, B lymphocyte differentiation to the plasma cell stage, and antibody secretion through the cytokines that the T helper 2 cells produce. So, to support the humoral response against the extracellular stages, like the merozoites and sporozoites, we need a Th2-supported humoral response. But what about the intracellular stages? There is very little that we can do to combat against the erythrocytic schizogony, the erythrocytic stages. The macrophages have to do all that work once those red blood cells lyse or once they're carried to the spleen or liver. However, we can see some cell-mediated immunity against the liver stages. So we know that CD8 positive cytotoxic T cells can kill Plasmodium-infected liver hepatocytes. Just to remind you that CD8 positive T cells recognize class 1 MHC plus antigenic peptides. So if these plasmodial-derived peptides are presented on the cell surface with class 1 MHC, then cytotoxic T cells should be able to bind and to induce apoptosis. One limitation, again, though, is that liver hepatocytes express only low levels of class 1 MHC. In fact, that's why liver transplants are one of the most successful transplants, because of low-level class 1 MHC expression. So the cytotoxic response typically is not enough to completely destroy the exoerythrocytic stages in the liver. It's only partial protection. The CD8 positive response is supported by the Th1 subset of CD4 positive T lymphocytes and the release of interleukin 2 by these cells, because interleukin 2 allows for the activation and clonal expansion of CD8 positive cytotoxic T cells and is necessary for their full activation to the killer state. Finally, the topic of antigenic variation. So we've talked about the fact that Plasmodium has a hide and seek escape strategy. We've talked about the fact that the multiple stages of Plasmodium are antigenically distinct. And we have mentioned the fact that some species of Plasmodium show antigenic variation. And this phenomenon has been studied best in Plasmodium falciparum. In Plasmodium falciparum, we know that there are about 50 different genes, designated VAR for variant genes. And these proteins encode proteins that end up being deposited in the infected red blood cell membrane. And these Plasmodium VAR proteins actually mediate the adhesion of the knobs, and these are knob proteins. Now, only one of these VAR genes is expressed in the population at a time, and these proteins are strongly immunogenic, so they do stimulate a strong response by the host. This strategy allows the parasite to persist in the host for a long period of time by continually shifting the VAR genes that are being expressed. So this is reminiscent of the African trypanosome story, with the exception that there's only about 50 genes, and that the repertoire then is more limited than we saw with African trypanosomes. The clinical immunity that builds up over time in endemic areas is thought to be related to increased exposure over time to these various VAR proteins, and may actually explain in part recrudescence in falciparum malaria. That concludes our lecture over the malarial parasites. And in the next lecture, we will talk about a few other examples of important human pathogens that are in the phylum Apicomplexa.